Thank you very much.
If you're feeling short of breath, you don't need oxygen either. Okay.
Well, it's really a pleasure to be talking to family, and I have a very interesting and exciting story to tell you about something you probably don't come across every day, and it has to do with the use of some what you might think are exotic materials. They're actually not exotic. These are materials that we're unfortunately too used to. For example, the nanomaterials that I'm referring to here, that we're going to be using to modulate the T-cell immune response, are made from material that you use all the time, suture material. And this is basically what we did. We figured a way to formulate these in very small objects, nanoscale objects, the size of viruses or very small bacteria. And the process of doing that enables you to actually also incorporate things on the inside, put things on the outside. What I've been focused on for the past few years is trying to use these materials in targeting the immune system, specifically T cells, lymphatics, dynamic organ, moving parts, and these are also moving parts. They're very small. They can get into different spaces, and so you can see where the correlations may be. But the question is, how do you, what questions will you be asking, and what are you addressing? So forgive this very simplistic overview of what I'd just like to align on. Make sure that we're all on the same page regarding what I'll be focusing on. The antigen-presenting cells, as you well know, macrophages, dendritic, professional antigen-presenting cells like dendritic cells, continuously circulating in the body looking for foreign things, pathogens. They take it up, and if they recognize it as foreign, process it into little bits and pieces, and then they migrate to local lymphatics, local police stations, where they present their findings to lymphocytes. And what's presented really is a bit or piece of that pathogen within the context of a molecule called major histocompatibility complex. Now, at that point, everything becomes kind of complex and very crazy to really keep up with. The dendritic cell can interact with a T cell a number of ways. For example, through recognition of the peptide MHC complex, a T cell receptor on a T cell would recognize that. If that happens with a phenotype called CD8s, those cells are induced to proliferate and differentiate. Daughter effector cells that can go out in the periphery and look for the pathogen that instigated this response and kill it. What can also happen is the peptide MHC can ligate to what we call also a TCR, but on a different phenotype of T cells, the CD4 positive T cells, and that leads to activation, what we call the helper response. Those cells help B cells secrete antibodies that tag the pathogen that started this cascade. The idea is to really try to flag the danger, the alarm. And of course, this is very simplistic, as I mentioned. But it's not just ligation of a peptide MHC, TCR. There are a number of other things that can happen in this talk. For example, there is something called co-stimulation, which I'm sure you're very well aware of, between a T cell and an APC. Lately, it's been co-stimulatory blockade has been a fascinating new way, introducing new exciting modular immunotherapies. There is also a talk, literally, in the sense of paracrine delivery or delivery of molecules like cytokines, interleukin-2, which activates T cells to proliferate. Now, the two T cells that I wanted to form this story about are right there, actually. The effector T cell response, simplified as CD8 cells, which are part of the effector response, the cytotoxic T cell response. And the other one that we're really all focused on as well, recognize very important, is the regulatory T cell response. And the proposition I'll make to you is that these nanomaterials that are made out of very basic building blocks and safe materials that we're very accustomed to using, formulated differently, can be used to modulate the direction, increase CD8 T cell proliferation, increase regulatory T cell proliferation, or perhaps the opposite, where it can wipe out regulatory T cells increase CD8s, wipe out CD8s, increase Tregs, and what do you do? And I think the answer to that question lies within this very interesting, fascinating, simplistic diagram, in that we need key signals to be incorporated in these materials. But is it really that simple? And that's what I want to spend some time talking to you about. The examples I'll give you, and I recognize I probably will not have enough time to go through all this, but at least I'll touch on each one, give you a flavor of why this is an important area right now emerging, is the use of these materials as vaccines. There are a number of questions to answer in terms of use of these materials as vaccines. Some of them are there. The encapsulated material that we're putting in is not really released all of a sudden. It comes out slowly. As you know, sutures dissolve slowly. And so if there's material encapsulated, the materials encapsulated will come out slowly as well. So the question is, antigen release, kinetics of antigen release matter. Pathogen-associated patterns may be a very interesting way to modify the surface, make these materials look like bacteria or small viruses in a safer format. Multiple PAMPs. Does it work on the human side? Can we target different human DC subsets? And actually, this is a very small subset of questions that we've been looking at over the past number of years. I will try to get to some of these. But then the other applications that we're really interested in is the total opposite of the vaccine, what we call the anti-vaccine response, which is the antigen-presenting cell is presenting a peptide MHC to the T cell, either CD8 or CD4, or other cells. The question is, can you actually make that immunostimulatory event into an immunoregulatory event? In other words, a tolerogenic dendritic cell instead of a stimulating dendritic cell or effector cell stimulating DC. And there's a very interesting way to do that, and it's actually quite simple. And then there's something that I think is a very good way to bypass the whole antigen-presenting cell format. As you know, Dendrion is a company that was marketing dendritic cells and has used them called Province. Fortunately, they had some financial issues. I don't know what their status is right now, but this is a kind of an alternative to cell therapy. Is that we're trying to create really the basic tenets of a cell within these particular formats, called artificial antigen presentation. How well do these work, and what can they be used for? So, just to rest assured first that the materials we are telling you about is not some academic curiosity. Actually, it's very far along in terms of clinical trials. What you're seeing here is an example of a evolution of how we went from small molecule discovery to focusing more on the delivery aspect. And this is just a sort of a simplified, simplified way of looking at that evolution. These particles are being made by Bind Biosciences. For example, I think they're in phase 2 something clinical trial. By the way, I should declare that I have no interest with them. I'm not part of Bind. But I skipped over a slide. Yeah, there. This. They are actually in, I think they're past phase 2, and they're actually proceeding beyond that. Results have been good. They have a number of things that are ongoing. And the safety by distribution, the basic questions we all ask about use of new things in our bodies have been addressed very completely, I think, with this specific nano platform. But the applications now is what really is gaining quite a bit of attention. So what are these? These, again, as I mentioned, made out of suture material, a type of suture material, I should say. It really is polylactic coglycolic acid, which degrades, degrades slowly in water, hydrolytic polymer over time, and it can be formulated as small particles. Now, the process of formulation is quite easy to visualize. If you think of vinegar and water and oil, and if you just take a water oil, we call a water oil emulsion, and you just shake that mixture very very actively, you get these little droplets, but if you leave it alone, it will then again phase separate. And the trick that we deal with, the challenge that we've been faced with is how to stabilize these droplets once they form, so that they don't coalesce and form these aggregates. There are ways of doing that, and they're very effective ways. And so during this shaking process, you're adding in essentially what you're interested in encapsulating, and at some point along the emulsion formulation, you are, when the material is hardened sufficiently, you're actually decorating the surface. So it's an interesting procedure, something that's founded on common sense, and seems to work well in terms of specificity and selectivity of the cells we're looking at. Advantages of these things are listed here. Obviously, they could be made very small, smaller than 15 nanometers, actually. The controlled release is one of the distinct advantages because, again, of the degradation of the polymer, which facilitates slower degradation or slower release of the encapsulated drug. And then you have targeting and retention. High avidity binding is one of the key features, actually, key advantages of these things. Higher avidity has been really a challenge for us in terms of when I was a graduate student, I worked with peptide MHC TCR interactions on looking at their interaction affinity on the using surface plasmon resonance. Very weak affinity interactions. If you're going to target the peptide MHC or even the TCR with its natural cognate ligand, avidity makes a tremendous difference in terms of off rate or dwell time on that specific cell. And so these are high avidity, like Velcro kind systems that can stick specifically to their target. They can also stick somewhat non-specifically. Another challenge we're faced with is how to control this selectivity. And I'm not going to bore you too much with the chemistry of playing around with these formulations. What I want to get into is sort of the applications, and again, really quickly, 3 examples of interesting applications related to immune modulation, specifically of T cells and regulatory T cells, CD8s and regulatory. So what you're seeing here is a picture of viruses and bacteria, small objects. What they have in common with what I'm talking about are those 3 things here. Small, high surface to volume. There's a reason why nature created small things. It's a very good infectious agent. It actually is the best way to facilitate both transport as well as intracellular and intracellular transport and extracellular binding. In the sense that you can put a variety of things on the surface. Again, you get high avidity, but you can also put a variety of things on the inside. And we were motivated by this for development of vaccines. And so a dendritic cell recognizes particles, small particles, recognize them very non-specifically, eats up these objects avidly, and then once they're uptaken, there's processing and the cascade of responses mentioned to you. The addition or the presentation of a pathogen pattern on the surface, of which a few examples are shown here, like lipoproteins, flagellin, lipopolysaccharides, really facilitates quicker recognition, not better uptake. It's really, it has to do with kinetics, and in terms of trying to therefore look like these effective pathogens, but also carrying a useful load vaccine, we were thinking about putting a PAMP on the surface. And the idea would be targeting toll-like receptors on lymphocytes. And this concept is actually, it seems to me like it's very new, actually, but it's actually, now it's pretty old. It's been around now for about, I'd say, 4 years or so in the biomaterials world, and it's evolving very fast, but it has advanced quite rapidly, and I think you'll see why for a second, in a second, because it works very well in terms of activating both arms of the effector response, the CD8 as well as the CD4. Now, what you're seeing here is a picture of dendritic cells that have been incubated with these particles. The particles are decorated with a PAMP. The PAMP is actually CpG, an oligonucleotide pattern. And so they have CpG on the surface. What they have inside is the West Nile viral envelope protein, which is what we're trying. There's currently no vaccine. The question we're asking is, will this be an effective vaccine against West Nile? Very straightforward question. So then straightforward experiment. Animals were immunized, subcutaneously immunized with these particles, and then the serum was collected, and a cohort of those animals that were immunized were challenged with the virus, and another cohort were analyzed in terms of what their immune response, what their T cell and antibody profile looks like. And this is the results that we get. In terms of survival, first of all, these CpG particles with CpG on the surface, West Nile viral envelope, these animals live longer. It's not 100%, but there's a number of other aspects, number of other parameters here, like for example dosage, etc. But the important thing is to compare it to alum, which is our current conventional vaccine adjuvant, and the difference is better. And then, of course, in terms of why this works very well, because look at the titers, the antibody response is increased, and the nomenclature here is CpG ODN, oligonucleotide / means what's on the inside is the West Nile viral envelope protein. You'll note that alum, or called also alhydrogel, didn't give you a humoral response. Surprising, given the fact that we use it as a conventional adjuvant. And the reason for that is because we were using the same amount of adjuvant, essentially total weight. So if you use more alum, you will get a bigger response. But of course, you get higher risks of other things, toxicity being one of them. The negative slash are particles with nothing on the surface but the West Nile viral envelope protein there. You get some response. And the other panel that you're seeing here is the effector immune CD8, CD4, 44. Positive cells are expanded preferentially with the CpG particles containing the protein, and the phenotype of those cells are actually memory-like. A memory-like phenotypic response that we see. There are so many permutations on now how you can do this experiment. For example, what happens if you add another PAMP in addition to the CpG? For example, what we added here, we put CpG on the inside, and we put lipopolysaccharide variant, actually an MPLA safer form on the outside, kind of like what a bacteria has. Extracellular ligands that are PAMPs and intracellular PAMPs. And that actually works better than either alone in terms of both induction of humoral responses, in terms of induction of a cellular immune response. I'm going to stop here with the vaccine story and just summarize really quick points. Points being that there is, what we notice is with these slow-releasing systems, you get persistence of a antibody. The antibody is actually present over longer periods of time with slow-release antigen availability. A depot effect. The other thing is that this is really not a material issue in the sense that any slow-releasing system will do this. We used PLGA here because it's safe. We could have used other materials. And then the most important take-home point is the activation of the CD8 and memory-like phenotypic response. And why is that? Well, because of a shortage of time, I'm going to just quickly skip over a slide here and show you this, because this really sort of explains how these systems work when they see antigen-presenting cell. A dendritic cell eats up a particle like it would any other particle, non-specifically. Again, the addition of a PAMP just enhances the kinetics of uptake. But then what happens next is interesting. In step 2, as the particle gets endocytosed, early endosomal, late endosomal compartments, what we know happens is that these particles tend to escape or leave those endosomal, lysosomal compartments. Where along that intracellular cascade does it happen, we don't know. I think a lot of people don't know yet, but there is a general understanding that these PLG particles escape the endosomal compartments. Once they escape, they get into the cytosol. During this whole time, by the way, they're releasing slowly because they're in a water environment, and as they're releasing now material in the cytosol, and that's priming class 1. In the endosome, they're priming class 2, which is the CD4, which gives rise to the CD4 response. In the cytosolic compartment, you're getting priming of class 1, which gives rise to the CD8 response. See, this phenomenon known as cross-presentation, very important not only for vaccine design, but also for cancer therapy, obviously. In terms of using tumor-associated antigens that we want both antibodies as well as effector responses that recognize those tumor antigens, and this is one effective way of inducing a response against them. I want to, along the lines of cancer, I'm showing you here a table. This is one of the questions I posed. A table of antigens, a tumor-associated antigen, cancer-associated antigen in general, are not a single, it's not one known, actually a whole panel. And myself and other collaborators here, including Dr. Dudakapar, which I'm not sure if he's here or not, we worked collaboratively over the past 3 years to try and answer the question, can you now use these things to encapsulate not just one antigen, but a whole host of tumor-associated antigens? And he had discovered a panel called the SOX2 panel. What I'm showing you here is a panel of infectious agent, influenza, EBV, CMV, but there is also another panel called tumor-associated antigens called SOX, comprising approximately 32 different peptides that were encapsulated, and what we noticed was you get a specific response against each and every individual one of those peptides. Actually, it wasn't us who did this experiment, it was Medhav's lab, Dr. Dudakapar's lab, and that was published I think summer of last year in this journal, Immunology, if you're interested. So I think the potential of using this system for tumor lysate, or perhaps just a panel of well-known antigens, cancer antigens, it's a viable product. It's no longer a curiosity. It really can work in that direction. So to summarize, this is a way to develop better vaccines, induce cellular and cellular as well as antibody responses. The other half of the coin, the regulatory component, is really interesting because essentially what we're trying to do now is tamp down the DC's ability to induce a T cell response. And the rationale there was, well, let's do two things. Let's encapsulate the antigen, which we've done before, but with it, because it's a combinatorial vehicle, can contain a number of things. An immunomodulator, for example, rapamycin. It's a immunosuppressive drug. And then let's decorate this with antibodies that will facilitate quick dendritic cell uptake, DEC-205, ligand to the C-type lectin receptor on DCs. And in this experiment, which is really interesting, actually, show you this because the results are actually were unexpected. There are 4 different configurations of this system. There are, again, nothing on the surface, an antigen, nothing on the surface, and then the antigen plus the drug, and then DEC-205 on the surface, antigen, DEC-205 antigen plus drug. And the experiment is simple. You take animals, you immunize them. 7 days later, you analyze their antibody response. Now, hopefully, if this really works the way you think it should work, you will get very minimal antibody generation or humoral response, OVA-specific antibody response. So this is the results of this experiment. The interesting part is actually not quite obvious. I'll point that out to you in just a second. If you look at the antibody response on the y-axis and then the different configurations on the x-axis, I think it's safe to say that the hypothesis turned out to be true. In fact, that the antigen alone, it stimulated T cells as was expected, and the antigen plus a drug actually suppressed the antibody response or generation in animal as expected. But what's really interesting is well, compared to soluble agents alone, you don't see the same kind of response. It's actually interesting. It's not shown here. It's shown here. Sorry. If you were to do this in a different, if you were to just take the antigen in one particle, the drug in another, combine them, mix them together, and just use that as your formulation, that doesn't do the effect. In other words, the combination of antigen and the drug need to be spatially localized to the same exact cell. And this experiment here shows that what I just said verbally, in the sense that this DEC-205 plus OVA particles, these are particles that are separate versus the combined. And what we're looking at is the OVA titer, and you can see there that combining them in the same system is very important. We really never gave thought to the idea of localizing to on a single cell level the antigens or the drugs, and actually, especially if we're thinking about combinations. When we inject combinations of things, we're hoping for the best. And what this is a procedure to really rationally direct that combination to the right cells or cells of interest. And then, of course, these, again, as before, we notice a cellular correlate with this antibody titer. In other words, you also get a minimal cellular immune response. Okay? In terms of the effector response that gets generated by these, the regulatory T cell response that gets generated, it's really very effective. So the phenotype we're looking at is FOXP3, CD25, CD4 positive T cells, which are the regulatory T cells that are stimulated by this dendritic cell presenting an antigen and containing a drug suppressor. So these are what we call tolerogenic DCs. And we're looking at here is the percent of those cells overall. And what you see is what happens when you mix agents just in a soluble fashion, as we normally do in the lab, compared to just those agents that are encapsulated. In terms of the amount of TGF-beta that is generated is exceeded. TGF-beta is, of course, a product of regulatory T cell hovering over microenvironment or inducing the stromal environment to generate TGF-beta APCs in that environment. So TGF-beta levels here are exceedingly high compared to just the soluble agent. And that's the theme that you see with these systems quite frequently. The amount of the response is elevated at lower doses and lower frequency. Okay, so how much time do I have? We're past the time. Okay, all right. So I think, I think this sort of, I wanted to delve into the B16 side, melanoma side, and apply what I mentioned to you here regarding melanoma. But I think you can put together the points, and there are a number of papers that have discussed this work. So thank you very much for your attention, and I appreciate your time.
Very good.